Hello everybody, we'll start from this slide. You can see the cerebrum, one hemisphere you can see in this picture. There is another hemisphere in the other side. So as you already know that the surface of the cerebrum has many sulci or grooves, and in between the grooves the ridges are called the gyri. So this picture you can see many shallow grooves. These are the sulci. Singular is sulcus and plural is sulci. So in between the sulci, the ridge or elevated area is called the gyrus. So many sulci or grooves and many gyri or ridges on the surface to increase the total surface area in the cortex. As you know, we already talked about this, that during prenatal and postnatal period, the number of neurons increase in huge amount, and that neurogenesis increase the number or population of the neurons, and to accommodate the increased number of newly generated neurons, you need more surface area. That's why the grooves and gyri are formed. However, something interesting happens when these grooves are formed. To form the groove on a plane surface, so in the very early stage the surface of the cerebrum is plane, right? Then gradually the grooves are formed. So to form the grooves, the cells must die. Probably you know that when the embryo becomes fetus, the hands and legs, those developed, so those are like birds, and then they grow and the fingers are get separated. So initially it is like one piece, and then many cells die there to form individual fingers in your hand and foot. Okay, so in these areas the cells must die, and that is called apoptosis, programmed cell death. So how these cells and which cells will die, those information um, is already programmed. So that's how the bifurcation takes place. Similarly, on the surface of the cerebrum, many cells die to form the grooves. So apoptosis takes place, and that increases the total surface area to accommodate new cells there too. So it is a very interesting process. Cells are dying, apoptosis and neurogenesis are producing new cells, and differentiation also takes place. What is differentiation? In the developing brain, the neurons of each area get their own properties. Like these neurons are, for example, for visual. These neurons are for auditory processing. So that is the differentiation, right? So after the neurogenesis, new cells are formed, and migration, then differentiation. And now if you see the cerebral cortex, as I mentioned before, there are many functional areas are present in the cerebral cortex. Just here you see only few. This is the occipital lobe here. This is the parietal lobe, the bluish cyan. This is the frontal lobe, big in human brain, and this is the temporal lobe. So there are 4 lobes. Occipital lobe in general is mainly for the processing of visual. So many visual areas are present in the occipital lobe. Okay, the temporal lobe is mainly for auditory processing. Okay, and the parietal lobe is mainly for somatosensory, and frontal lobe, this one is bigger in the human brain, and the frontal lobe is mainly responsible for the motor functions as well as higher order brain functions. So what are the higher brain functions? Higher brain functions are the functions those are uh, more related to cognitive processing, like thinking, um, judgment, emotion, analysis, problem solving, right? Doing math, critical thinking. Those functions are considered as higher order brain functions, and frontal lobe is mainly responsible for those functions. Now you know that lower brain functions include micturition, parturition, defecation, right? Those happen to everybody, like in higher animals as well as the lower animals. So those things happen, like urination or micturition, defecation, those things, those reflexes, those are considered as the lower brain functions. So what happens, as I mentioned, that the occipital lobe is for the visual, and all the way in the back, this is the primary visual cortex. Okay, and then you have the secondary visual cortex and other visual areas. From the occipital lobe, visual signal goes to the temporal lobe as well as to the parietal lobe. So visual signal also travels to the parietal lobe as well as to the temporal lobe. Okay, we'll talk about these two pathways later when we'll talk about the visual system. Okay, and you see, this is the lateral sulcus. It is a deep sulcus, and this area is called the primary auditory cortex. So this is the primary auditory cortex at the bank of the lateral sulcus in the temporal lobe, but near the lateral sulcus. Okay, and in the frontal lobe you have two special senses, cortices for two special senses. Primary taste cortex is here. This area is also called insula. Okay, and in the inferior of the frontal lobe, this area is called the olfactory area, primary smell area. You see here, this line here, this is called the central sulcus. This sulcus is important. Why? Because this central sulcus is separating the frontal lobe and the parietal lobe. These two lobes are separated by the central sulcus. Okay, and what is important here, that in the front of the central sulcus, this gyrus, you see pretty prominent gyrus, long. This is the primary motor cortex. Okay, and behind the central sulcus, this blue one, this prominent gyrus is called the primary somatosensory cortex. Okay, so central sulcus separates these two functional areas. Okay, there are many other functional areas are present on the surface of the cerebrum. There is an area here This is called the Broca's area, named after a French neurologist, Broca. And he had a patient who he got a stroke, and then he lost his speech ability to speak. And after his death, his doctor Brocas, he dissected his brain and examined and found that this area was lesioned, damaged due to stroke. So he first proposed that this area is responsible for the speech production, and this area is the Broca's area. Okay. Then if you see in the back, partly in the parietal and partly in the temporal lobe, there is an area here. This is also related to speech, but this is not speech production. This is more related to speech recognition. Speech recognition. You hear my cat is making sound. Okay, he's playing. Anyway, so speech recognition, and this area is called the Wernicke's area. Areas both are related to speech, but in the frontal lobe, Broca's area is for the production of speech, and in the back part, Wernicke's area is for the recognition of speech. So if lesion occurs in Wernicke's area, the person will be able to produce speech, will be able to talk, but will not be able to understand what other people are saying or even what he is saying. And if the Broca's area lesion occurs or damage occurs, then the person will not be able to produce speech, but will be able to understand. Okay. Important principles of sensory computation. So these are some important principles. One is the size of the receptive field. It is important. Then sensory systems are topographically organized. So you will see nice, map, systematic map present in the sensory areas in the brain. Hierarchical and parallel processing take place along the pathway. We'll talk about this. Temporal, spatial population and frequency codings take place along the pathway in different areas. Ascending, that is bottom up, and descending top down processes interact, or pathways interact each other during the transmission of the sensory signals. And convergence and divergence take place along the sensory pathway. Okay, and another one is the signal to noise ratio. That is important determinant of the sensory perception. So these are some important aspects of sensory computation. First, the size of the receptive field. Each receptor, sensory receptor, has its own receptive field size. Okay. For example, these are the sensory receptors or receptor cells. For this one, the size of the receptive field on the skin, tactile receptive field size, is like this. Okay. And you see the dotted line is showing the size of the receptive field for each receptor or neuron. Okay. And one thing here, you can see that these receptive field sizes are same. However, that is not true everywhere. If I measure the skin surface on the back of your hand, then these are the sizes. Okay. For example, now if I go back to your fingertip, you will see the sizes are much smaller. Okay, and the number of neuron or receptor is much higher. So again, if I measure here, the receptive field size is much bigger, right? And each neuron is getting signal from each surface area, part of the surface area. So these are the receptors or neurons, and so these are the neurons or receptors, and these are the receptive field on the surface. Make sense? Now if I see the fingertip, then what I'll see that the size of the receptive field is much smaller, right? So each receptor has much smaller receptive field size. That means what? From each area, one receptor is getting the signal. Make sense? That tells you that in the same sized area, you have more receptors in the fingertip. So this is fingertip and this is back of your hand. Okay, back of your hand. So you can see that since the surface size of the receptive field is bigger in the back of your hand, the number of neurons in same sized area is much lower than the fingertip. The number of neuron is much higher because their receptive field is smaller. Make sense? Now, different areas have different amount of or density of receptors, and where the density is more, the size of the receptive field is smaller, and where the size of the receptive field is smaller, the perception is better. Okay, better perception. Okay, then this one. Now let me give you another example. You know that in the retina of your eye you have the photoreceptors. Now if you see the center of the retina, you see the cones. The photoreceptors are present in very high density. Why? Because the size of the receptive field of those cones is very small. Okay, so in this small area you have huge number of photoreceptors, okay, in a smaller area. And as you go outwards, the photoreceptors are gradually less in number. Okay, that means what? The center part of the retina has highest acuity, the ability to see, right? So you see where the number one, the number of receptors are not same everywhere, and where you have smaller receptive field, in that area you have more receptors and the perception is better. Makes sense? So that is the location and the size of the receptive field. It is like on the TV screen you have the pixels, and you know that in 1 unit of time in which TV you have more pixels, the number of pixels, the clarity is better, right? The picture is more clear. And if you have the TV that has less pixel, then the picture is less clear. So it's like TV pixels that you all know. Changes of the receptive field size at different levels. So we see that as the sensory signal moves upwards towards the brain, the size of the receptive field increases. in the neurons. So this is more like you see, this is your retina eye, and from the retina, when the signal goes to the thalamus, and here then from the thalamus it goes to the cortex. So let me just think about these three areas. Okay, so this is the object, the whole picture of the object. When it falls on the retina, retina does what you see. Let me draw here more clearly. So this is the object, whole picture, and when it falls on the retina, retina breaks the entire picture into many tiny pieces. So that's the function of the retina. It breaks the picture into many small pieces, and the receptor, each receptor will get one piece, and then these pieces from the receptors are sent to the higher areas. So when it is sent towards the to the thalamus, for example, you will see that the pieces are joined together. Okay, so each neuron has larger receptive field. So less neuron is needed here than here. Makes sense? Then as it goes to the visual cortex, all these are joined together, okay, to give you the complete picture. So you see that the size of the receptive field of the neurons increases. Makes sense? For example, how that happens? That happens due to convergence. For example, these are the retinal receptor cells. Okay, all these are neuron type cells. Okay, and each is processing like one dot. Then, for example, these two are sending signal to one thalamic neuron. Make sense? These two are sending to another thalamic neuron. Okay, these are sending to another thalamic neuron. So you see the convergence. So since this one is getting from 2, so its receptive field size will be double. Make sense? And now if these two are joined together in a neuron in the cortex, then it will be much bigger. Make sense? So that's how the convergence takes place, and the size of the receptive field in the neurons gradually increases as you go towards the brain. Okay, and that's why we say that earlier neurons are more simple type neurons, but the higher areas neurons are more complex neurons, because multiple receptive fields joined together, are combined together as you move upwards. Okay, so that is another observation or another property we see as the sensory signal travels towards the brain. As I mentioned already, I explained this, the density of sensory receptors and the size of the receptive field, both are important, and if the number of the receptors is more, the size is smaller, the perception is better. Okay, hierarchical and parallel processing. Two things happen. One I already explained. As the signal you see shown by the dark line goes up, it becomes more and more complex, right? Because the signal is moving from the simple neurons to the complex neurons. Okay, and more signals are being combined together, more information is being combined together. Okay, for example, when you see an object, for example this object, it is rectangular, okay, and the color is red. So red color. And now initially from the retina, the color, the angles, the shape, right, texture, all those will be processed separately. Then as the signals will move up, these information will be combined together. Okay, so these neurons are more simple because they are processing single type of modality or function, but as you move up, the neurons get more complex and multiple factors are being combined together and being processed in a single neuron, right, more than uh, the earlier neurons. So that is the hierarchical processing, okay, that I explained before. Now parallel processing is also important because, as you know, that when the signal from any um, object moves towards the brain, uh, multiple information travel to the brain, and you already know on the surface of the brain you have many functional areas that I explained at the very beginning today. So for example, this is your primary somatosensory cortex, and this is your secondary somatosensory cortex. Okay, so this is S1, this is S2. Okay, so when you touch something, that touch, like if I touch a piece of metal, a number of information travel towards the brain, right? And for example, if it is cold or hot, if the surface is smooth or rough, right? If it is soft, the object is soft or hard. Just I am giving you 3 examples. So you see multiple information we gather from each action when we touch the surface of this object. So some of those signals are taken to the primary somatosensory cortex to give you an immediate perception about that object. Some signals are taken to the secondary somatosensory areas. So there are multiple areas, right, where this information is being taken and processed. So these neurons will process some functions, these neurons will process some functions, and these neurons will give you immediate perception, then more information will be given by these neurons. So what happens, you see, this is a single nerve that is taking the signal from the skin to the central nervous system, right, to your spinal cord, and from the spinal cord the signal is traveling upwards, okay, through the spinal cord. And you see here two nuclei, gracillae and cuneate nuclei, and the signal here, you see the nerve is divided, right, and some signal is being processed in this nucleus and some signal is being processed in this nucleus. So the bifurcation, and this is termed as primary bifurcation. It makes sense, right? Primary bifurcation. And then also what happens, we see it's not shown here. From here, for example, it divides again to go to 2 separate areas, and in this case we'll say secondary bifurcation. Make sense? So in the ascending pathway, the nerve divides into 2 first, and these are primary bifurcation. This is primary, first division, bifurcation, and then signal goes to 2 different nuclei. Then from there as the fibers carry the signal upwards, they can again and again divide into more and more branches, and more than two areas can process the signal like this. So that is called the parallel processing. Okay, this one is processing some signal, this one is processing different type of signal, this one processing another type of signal, this one is processing another. Okay, then they will be taken to the cerebral cortex. Make sense? So along the ascending pathway we see this kind of parallel processing. That means the bifurcation of the nerve or fiber takes place. Divergence and convergence along the ascending and descending pathways. So let me give you an example of divergence and then an example of convergence. Both are important and happen or take place along the pathway. So this is your skin, and if a stimulus, for example touch, stimulates the receptors, these are the receptors here, and this stimulus has activated these three receptors, all these three receptors. This is a model. Okay, and since the touch is here, this receptor will be activated, the middle one will be activated more. Makes sense, right? And these are a little bit away, so they will be activated less, but all these three receptors are activated. So you see, this one will produce more firing or action potential. Okay, respond more than these two, less, but all three are activated. That's why you see the firing. Okay, now the signal from these receptors, when the receptors fire, the signals will be taken to the higher neurons, right? So here all these neurons are getting the signal from the receptors. Okay, now you see here something, this is called the divergence. So from this middle receptor, let's see the middle one, from this middle receptor, when the signal is going upwards towards the higher neurons, you see the fibers or nerves divide. Okay, here you see three divisions, right? So all these are excitatory. So this one will be directly getting, this one is getting a signal, as well as this one and this one. So these three excitatory inputs are on these three neurons. Okay, so they will be excited. However, you see, when the signal from this one will go to the higher neurons, also it is divided into three, the fibers, and this one, the middle one, is also getting excitatory input from this one, and similarly this one is bringing excitatory signal here. Okay, so the middle one is getting excitatory signal from the center receptor, as well as excitatory from the other two neurons, other two receptors. Make sense? So how many excitatory here? Three excitatory input, right? So this one will fire more than this one. Okay, because it is called summation. Three excitatory inputs. Similarly, this one is getting from this one is getting from this one. Makes sense? And this one is also getting excitatory input from the middle one like this, and this one is also getting from the middle one. So this one is getting 2 excitatory, this one is getting 2 excitatory, right? One from this one from this one. This one is getting one from this one, another from this one. So the firing will be, also in these neurons. So you see, as you move upwards, divergence, that's how the divergence occurs. Branching take place, and more neurons get input from the smaller number of neurons. Okay, and more neurons are recruited that way by the divergence action. Okay, that happens. So you see here, 5 neurons here are getting excited. Okay, getting signal from 3. So this is a model how the divergence take place. Okay, so this one is getting 1 excitatory input, this one is getting 2, this one is getting 3, this one 2, this one 3, 1. Now also we see that convergence take place. So divergence and convergence, they are opposite. Convergence is from multiple receptors or neurons to fewer neurons or receptors. So just opposite of divergence. Okay, and for convergence, the inhibitory interneurons, you see this black shown by these black circles, these are inhibitory interneurons, they are activated. So something interesting here, you see the top part is same as the upper one. So here it is same, right? So the neurons are getting signal excitatory inputs from the receptors. So 1 excitatory, 2 excitatory, 3 excitatory, right? Then 2 excitatory, 1 excitatory. However, when these neurons are getting excited, you see the signal going down and coming back via the inhibitory interneurons. So let me just tell you, this is excitatory signal going to the inhibitory interneuron. So if you see this inhibitory interneuron gets excitatory signal, then the inhibitory function will be excited. That means it will be more inhibitory action. So now from this neuron, when the inhibitory neuron is excited by the excitatory inputs, you see here excitatory inputs on the inhibitory neuron, so inhibitory neuron will be excited, right? So inhibitory signal will go to the earlier neurons, these neurons. So here now you see the middle one, this is the middle one, got 3 excitatory and 2 inhibitory. So these 2 are inhibitory, these 3 are excitatory. These are coming from inhibitory interneurons. Okay, so this one is more positive, excitatory, so you'll see some firing. Okay, the middle one. But now if you see the next one beside the middle one, this one, it is getting 2 excitatory, however also getting 2 inhibitory. So these 2 are inhibitory, these 2 are excitatory. Makes sense? So they will cancel each other. So quite no action potential or firing. Same thing here, no action potential firing because 2 inhibitory, 2 excitatory coming from the receptors. So only you'll see firing in this middle, the center neuron. Makes sense? So that's what you see here. So this is convergence. So this is divergence. Many neurons are getting activated, and this is convergence. Less neurons are getting activated than the number of receptors. Okay, and In some cases we see divergence, in some cases we see convergence, and both are very important to process the sensory signal. Topographical organization. As I mentioned that in different sensory areas you will see the neurons are very systematically organized. What does it mean? This is a chunk of area empty in the visual part of the brain. So in the visual part of the brain, the occipital lobe, you have an area, small area that is called middle temporal, empty. If you see the neurons, the response of these neurons in this area, you'll see that neurons respond very systematically. Neurons are organized systematically. What is that? These neurons are sensitive to direction. So when you change the direction, you will see that a gradual change. For example, this is the empty. Okay, I'll see that if I move like 100 micrometer and record from these neurons, if I see that these neurons like this direction, then if I go here, you won't see just opposite, you will see slightly different. If I do this neuron, I'll see again more, but gradual change. If I record this, I'll see like this. So you see that systematic. It is not like this way and next neurons are just opposite. No, not like that. So very systematically organized. And you probably know, you will learn more in the auditory cortex. If this is the primary auditory cortex, we see belts, frequency belts or bands, and in these frequency bands, if you record the neurons, you will see that neurons are very systematically organized. Like if these neurons respond to low frequency, and these neurons will be the highest frequency, sensitive to highest frequency, and these will be medium frequency. So gradually from low to high or from high to low. It is not like low then very high then medium, not like that. So these kinds of systematic organization we see in all sensory systems. Okay, and I just gave you two examples, visual and auditory. Sensory coding. Coding is very important. Sensory coding follows a number of types of codes. One is called the temporal coding, another is spatial, then population coding, and frequency coding. These are some important sensory codings or codes. Temporal is very important. Just a simple example, like when you go to see the orchestra, many players, right, musicians, they play together, but all of them must play in a synchronous way, and that will give you a very good feeling, right, of music. If different people start to play without following each other, without any synchrony, then you won't enjoy it. That will be kind of noise. However, you see so many people play, but that doesn't sound like a noise because they play in timely manner, maintaining that temporal codes. Similarly, your neurons, many neurons work together, right, but they must maintain the timing, okay, when which one will produce action potential, and that is the temporal coding. Spatial codes, you know that if I see the inner ear. Inside the inner ear cochlea, you have a basilar membrane. Okay, this is called the basilar membrane, and on the basilar membrane we see that the receptors are very systematically organized from high to low. So this is the tip of the basilar membrane, and these receptors are stimulated by very low frequency sound, and these receptors are stimulated by very high frequency. These neurons are stimulated by medium frequency sound, right? So you see that if these neurons are activated, brain will listen high frequency. If your brain receives signal from this part, it will perceive medium frequency sound, and if from this area, low frequency sound. So that is the special coding. So which area is being activated, the neurons or receptors of which area is being activated, that will give you the perception. Okay, variation of perception. So that is the special coding. Just an example, there are many examples. And population coding, if the stimulus is strong, this is a strong stimulus and this is a weak stimulus. Okay, this one strong stimulus will activate more neurons or more receptors. Okay, and this one will activate only few, more localized. Okay, so when more neurons are activated, usually the strength of the stimulus is, or perceived strength is more. Okay, so you'll perceive the stimulus stronger. So this is strong. This is when less neurons are activated by the stimulus, that gives you weaker perception. Okay, so that is the population coding, how many neurons are being activated. Okay, and another is frequency, how first we present the stimulus. Okay, if like I present 20 stimulus briefly in a brief 20 stimuli in one brief stimulus, and another is like this 20, but the frequency is low. Here the frequency is high, right? So this one will give you stronger perception than this one. Makes sense? So frequency, how many in one unit of time, that is the frequency. And if the frequency is high, usually more neurons are activated, and that gives you better perception. So all these codings are important in the neurons when the sensory signal is being processed, and that determines how you perceive the signal. Okay, so we already talked about this. Adaptation, we talked about the adaptation before, and two types of neurons, right? We talked about these two, fast adapting and slow adapting neurons. So rapidly adapting and slowly adapting neuron. And remember that a rapidly adapting neuron is more useful to tell you when the stimulus arrived, because the neurons, rapidly adapting neurons, briefly fire when the stimulus arrives, then gets quiet. So we can tell that a stimulus arrived here at this time. However, slow adapting neuron, you see the response gradually decreases. However, these neurons continue to fire as long as the stimulus is present. So you see, this is the stimulus it was present from here to here, that means all through, but this neuron just briefly fired when the stimulus arrived. So arrival time can be better detected by this one. However, how long the stimulus was presented, that cannot be detected by this neuron, that can be detected by this neuron, as this neuron continues to fire, although in slower rate, but continues. When the stimulus will be stopped, then it will not fire anymore. So you can tell that how long the stimulus was present by this one, okay, and this one when arrived. So this is like more arrival time, and this one is the length, how long it was present. Okay, both are important, right? Okay, so I'll stop here, and in next lecture I'll talk about the auditory stimulus and the anatomy of the ear.